Okay, so here we go. So my name's Dave Mason. I work at the Centre for Cell Imaging, which, as Alan mentioned, is a part of this overarching facilities management system called the Technology Directorate. So at the Centre for Cell Imaging, we're an imaging facility that supports scientific research at URL. And this really goes to UL. This really goes to the heart of.
Too dark, Dave. It's very bright at the back. And I know it's going to.
This really goes to the heart of what the TD exists to support, which is that if you put all the expertise and the equipment in one place, then people all the way from across the university can come and they know that they're getting the best possible service. I say beyond as well because we have lots of people coming into the facility from areas around the UK, especially sort of Northwest and other Merseyside, not only academically but also from industry. So what do I mean when I say imaging? Most of you are probably familiar with the idea of cameras. Maybe you've got a digital SLR. Almost everyone who has a cell phone has a little camera inside it. And what we do is not that different. We use microscopes instead of cameras to take the pictures, but the very underlying principles are actually the same. So you need really three things. The first is a light source. So if you're using your cell phone camera or using a digital camera, you can use the sun, you can use a flash. With our systems, we tend to use lasers, a little more high-tech. Lenses are basically the same. They're pieces of glass that can magnify a light source. And in the digital camera world, you maybe have 3 to 5x magnification. We tend to go higher magnification, sort of 6300x magnifications, and I'll show you some of that later. And then finally, you need a detector. So before most of you were born, people were acquiring images onto 35 millimeter film. These days, most of the cameras use CCD chips, so the digital cameras instead of analog cameras. You can also use an eye, of course. If you're using a microscope, like this one shown here, which you may have seen in some of your classes, you just look at what you're looking at. You can also collect things onto a digital camera, and I'll show you that most of the images we use are collected onto cameras. So this is what one of our microscopes actually looks like. It's a little more complicated than the little lab bench microscope I showed you in the last one. But even so, it has those three key parts. So I mentioned lasers before. So this big box that sits on the bottom that we'll see this afternoon is the laser box. If we look for the lenses, we use objective lenses. And again, these can go from a range of about 2.5 times magnification all the way up to 100 times or so magnification. And then the detector in this case is a scan head. So this just sort of bolts onto the side of the system. Now we have these three components. So you think, well, okay, this isn't that different. Maybe you pay £100 for a smartphone or a digital camera. These systems tend to cost in the order of about £600,000, up to £1 million maybe, depending on the specifications that you want. And what we're paying for with this quite considerable difference in cost are really three key attributes. So we need sensitivity, which is detecting very dim signals. In some cases, this can be down to even single photons, single packets of light. We need magnification, so we need to make really, really small things like cells and viruses and other things appear much bigger. And we need resolution. And resolution is a really, really key concept in the field of imaging, but it basically just means being able to separate very close objects. So if two objects are not resolvable, it means you can't tell there's two. They're too close together. If you move them further apart or you zoom in, the two objects become more spaced apart and you have better resolution. So these are the three things that we get from our expensive sort of microscope setups. So I'm going to give you an idea of scale, just so you know roughly what we're dealing with here. Each object I'm going to show you is going to be 10 times smaller than the last. So if you start off with a professional basketball player, something in the region of 2 meters, 10 times smaller, maybe a football, give or take. 10 times smaller again is maybe the thickness of your thumb, 2 centimeters. If we go 10 times smaller again, we're talking about a small insect, maybe a flea. That's 2 millimeters. So we're already 1,000 times smaller than our basketball player here. So if we keep going, you get to about the thickness of a human hair, 200 micrometers. And then we go down to cell levels. Cells vary hugely in their size, but 20 microns is a good estimate. 10 times smaller again, and we're into bacteria. So now bacteria are about 2 millionths of a meter. So this is a million times smaller than our basketball player. So basically, if you stack up 1 million bacteria end to end, it'll be a very thin tower, but it'll be about the height of a basketball player. So 10 times smaller again, and we're into viral particles, so things like HIV. 10 times smaller again, and we're talking about the thickness of a lipid bilayer. So this is the plasma membrane that surrounds most of the cells in your body, about 20 nanometers thick. And then the last one I'm going to talk about, or just mention today, is a DNA strand, which has a rough width of about 2 nanometers. So the 2 nanometer DNA strand is about 1 billion times smaller than our basketball player on the far side. And the region that we deal with in light microscopy sits about in the middle of this. So everything from small organisms, like the little fly here, down through the hundreds of microns to cells, bacteria, and in some cases some viral particles. But we're really limited to about 200 nanometers for the most part. If you want something smaller than that, you tend to go to structural or electron microscopy. If you want something bigger than that, you go to preclinical imaging, so things like MRI, X-ray, CT, that sort of thing. Okay, so the talk was imaging in 5 dimensions. So basically what I'm going to do, through lots of pretty pictures and what I consider fairly exciting science, show you some of the projects that are going on at the CCI. And we're going to go up through the dimensions, some of which you may seem obvious to you, some of which may not so much. So this is actually a fluorescent image. I'm happy to talk about the technology over lunch or in the tours this afternoon. But this is an image of 2 cells you can see here, labeled for something called tubulin, which is a cytoskeletal component. So this is the skeleton that gives the cell shape and allows it to move and so on and so forth. So these are our first 2 dimensions, X and Y. So most of the imaging we do, you probably don't even ever think about the fact that your 2D photographs are in 2 dimensions, but there you go. And this is quite nice. So it's a nice image, but we're going to move on and get a 3rd dimension in. The obvious one to go for next is Z, but for reasons I'll come to afterwards, we're not going to deal with Z until after this next one. So the next dimension I'm going to talk about is actually channels. So when we take an image like this, what you're seeing, the brightness, the fact that it's in grayscale, can just be thought of as an intensity. So it's an increasing number of photons. So in the dark areas in the background there, you can see that there's not very many photons, it's very dark, but in the really bright areas in the cell, we're getting many, many more photons. Now the important thing is that photons don't intrinsically have a color. The wavelength of the photons, so the way in which they move, dictates the color that we detect, but the photons themselves are discrete quanta. So what happens is if you try to do labeling of 2 different things, you'd end up looking like this. So in the first case, we have these lovely filaments. In the second case, there are all some blobs, but they're all overlapped because they're all photons. So a lot of what we do is actually separating these 2 different probes optically. So within the microscope, we can separate 2 different types of what we call channels. So in this case, what we can do is to label one in one color and one in another color. It's important to remember that these are all photons, and the labeling is actually fully artificial. So in this case, we've labeled one set in green and one set in red. So now we can tell them apart, and we can say how close is one to the other, and look at distribution and so on and so forth. And we can keep doing this with as many probes as we fancy to some extent. So in this case, I've labeled the DNA in the cell in blue. So as I said, it's completely arbitrary what color you pick. So the same image is now displayed in a whole bunch of different, and in fairness, quite ugly different colors, but they're all just different ways of separating the photons that are associated with one organelle, in this case in the cell, than another. So those are our channels, and it really adds the one of the 3rd dimensions to our imaging. So why don't we talk about Z now? So most people are familiar with Cartesian space and X, Y, and Z. This is actually the brain from an embryonic fruit fly. So these are the things you see buzzing around your fruit bowl in the summertime sometimes. So the embryos have been dissected and the brain has been taken out. You can see there's already some colors associated with it. I'm not going to go into the details of that for the time being. And this is a lovely slice. You can see there's some green patches, there's some red blobs, there's some blue blobs. You can really get a lot of information from this. But then you start asking questions, well, this thing has 3-dimensional structure. So how do we know if something is above or below something else? And it's a bit like Orion's belt. So when you look at Orion up in the night sky, all the stars look roughly the same brightness, so you assume they're in a flat plane. But actually, they're millions and millions of light years behind or in front of each other. It's just the angle from which we're viewing that makes them look flat. So what I'm trying to get at is that if we take a single slice, you get some information, but you definitely miss some. So what you can start to do is actually move up and down through the Z range and take lots of different slices. And the cool thing about this is that when you start to do Z slicing through the image, you all of a sudden get a lot more information about the 3D structure. So this is exactly the same sample, but now I've made a movie that just slices up through the section. And you can hopefully see that you start to get localization that different changes, and also you can get more idea of the structure, for instance, of the green parts. Now this is all well and good, but the really cool thing about it is that once you have a 3D stack, you can then render it in 3D. So you can make a fully 3D model of these, in this case, embryonic brains. And I've just made a movie here rendering in 360 degrees. But of course, at this point, you can also zoom in. If you want to look at a particular spot, you can go right into that. You can rotate it around. It's very futuristic when you actually try and manipulate these things. It's a very, very cool way to sort of visualize and get inside your data set. So that's one of the real advantages of having the Z dimension. So the final dimension I'm going to talk about is time. And in some ways, this is one of the most exciting dimensions to be imaging in, because biology really happens in time. If you take a snapshot of anything, you really, really lose the context of what's happening. And so a lot of the work that's done in the CCI is actually done in live cells and in time-lapse imaging. So I'm just going to go through a couple of examples. This is one, and it only uses X, Y, and time, so we don't have multi-channel on this, and we don't have a Z stack. The reasons why will become evident in a minute, but these are cardiomyocytes. So these are a type of cell that make up the smooth muscle of your heart. And the fascinating, if slightly creepy thing about these cells is that when you grow them in a dish in laboratory conditions, they will actually start to beat spontaneously. So we're not providing any electrical current here. It's just the fact that the cells have grown so that they're touching each other, they're making these boundaries, and chemically they're communicating with each other to make this beating pattern happen. And it's amazing. So this is actually captured at 30 frames per second, so it's quite a quick acquisition, and it's played back in real time. And this highlights one of the limitations we can have, is that if you want a very, very fast acquisition, so 30 frames per second, you really need to sometimes trade off doing multiple channel acquisitions or doing a whole stack of Z, because actually it takes too long to do all those acquisitions through Z, so you wouldn't be able to get the sort of speed of acquisition. And in this case, people were only interested in studying a single plane through the middle of the cells. So part of what I do at the CCI is also image analysis. So that's taking these lovely images or movies and actually turning them into information. And this is an example of doing that here. So what we're doing is actually measuring the contraction of these cells in culture, and then just drawing off, in this case, this is a displacement graph, so you can actually see the effective beating of the cells. The nice thing about this is that you can add drugs in, and it can have real clinical relevance when people want to start looking at things to speed up heartbeats or slow down heartbeats, and you can test them in cells without having to go to animal models. So this is another thing we've been doing relatively recently, is actually looking at spheroid models. So this is actually imaging of, this in this case would be X, Y, Z and time. So we're imaging through this thing called a spheroid. And a spheroid is about 0.5 millimeter in size. And imagine a solid lump of cells. So it's a big ball of cells that exists in a gel. And what happens is that when you get a solid tumor in your body, you often get something called metastasis happening, which is where cells are breaking off from that solid tumor and they'll go and lodge elsewhere, maybe in the lungs or the brain or in other vessels. And studying how this metastasis happens is really, really important, because then if you can know what the mechanism is of the migration, you can start to alter it maybe eventually in patients and reduce the rate of metastasis. So this is just a 3D rotation of a ball of cells. So this is one of the spheroids I was talking about. So this is the X, Y, Z that's just rotated. The neat thing is we can start doing this in time. So this is not rotating, but this is over 24 hours of imaging. So a whole day imaged every 10 minutes or every 30 minutes or so. And you can actually watch the cells migrate out into the surrounding gel. So this is it's a model, but it's a direct representation of what's happening when the cells are migrating out into the surrounding area. And it's really, really neat to play it all back afterwards. As I said, this is 24 hours of imaging compressed into about 30 seconds or 40 seconds. So again, coming back to what I do, which is largely image analysis, we can take something like this and we can actually follow in three-dimensional space and time where all of these individual cells are going. So all of a sudden it turns from a pretty movie into something that's highly quantitative. So we can actually start to track in 3 dimensions over time where these cells are moving. And I've just labeled a subset of the migrating cells here in yellow blobs with purple, sort of cut straight down the middle. So from this, we can really get a lot of data about the speed they're moving, the direction that they're moving, even just sort of the rate at which the tumor is expanding or the cells are expanding out into the surrounding media. So that's really where I wanted to end. I'm just going to sort of wrap up by saying that we use microscopes to study biological systems in real time and in multiple dimensions. We work, despite being called the Center for Cell Imaging, we work across the gamut, all the way from sort of virus-sized things or even single proteins in some cases, all the way up to whole but small organisms. So if you want some more information, we've got a load of information about the equipment that we have on our website, so it's cci.live.ac.uk, or feel free to drop me or the CCI an email, and I'd be happy to take questions now and also during the tour, or I'll be around at lunchtime as well. So thanks for your attention.
Thank you, Dave. Fantastic. Any burning questions? We're a little bit late, but if you're really, really desperate, you will get lots of chances to see Dave later.
In the absence of any others, just one quick one. These light microscopy techniques, there's no equivalent in electron microscopy, I take it, so there's no way you can get time dimension in electron microscopy, nor is there any sort of channel option in electron microscopy.
That's roughly true. So with electron microscopy, instead of using photons to illuminate your sample, you're using electrons. So you get a much, much higher resolution, and as such, you can image much, much smaller things. So the dimensions we can do are X and Y, obviously. We have a technique at Liverpool where you can actually do Z. So you can actually take a block of tissue, image it with an electron microscope, and then slice a block off slice off the end, and then image again, slice and image. So you can do 3D tomography with electron microscopy. We're really proud of that technology here at Liverpool, but not in the light microscopy facility. Channels are an interesting one, so you can use different sized gold particles to label different components of your sample. So the way electron microscopy works is it shows up signal that is electron dense. So if you have something, say some gold particles inside your cell, they'll appear very, very dark in an electron microscopy image because they're absorbing or scattering the electrons that are used to illuminate them. So by using different sized gold particles to label, say, the nucleus or the cytoskeleton, you can, in fact, to some extent, do different channels.
That's a really interesting emphasis in the course, the comparison between light and electron microscopy. You might think electron microscopes are cooler, they're higher resolution, but they can't do these things, which is why there's always going to be a place for light microscopy alongside.
Thank you. Thank you, Dave. Tremendous. Feel a bit